My name is Colin Daniels and I'm the Working Age and Young People Service Manager. So we sort of created a bespoke service for people who are just a bit younger, may not be with AMD, and the service is growing from strength to strength. I'm always at the end of the phone or emails and bits and pieces to answer any questions you may have around anything around living with macular disease, really, but from more of a sort of a working age point of view. Anyway, so that's sort of the introductions done. As I say, if you can just sort of leave yourself on mute and your camera's turned off, and I'll hand over to Rachel. So good evening, Rachel.
So hi, and thanks to Colin for that introduction and for inviting me to speak to you all today. I'm here to talk to you about some research that I've been conducting in recent years with the support of the Macular Society into understanding early onset macular degeneration. So I thought I'd take you through some of the basic biology that underpins a healthy macula and also what happens when the macula degenerates. And then I thought I'd talk about some of our early research successes on this topic before telling you about some stem cell models we've been developing and a new genetic test for this spectrum of conditions. And then I'm going to discuss some of the more important research questions we're trying to answer before finally talking briefly about some treatments that are in development and why a genetic diagnosis is important in that regard. So for vision, light enters the front of the eye through the cornea and the lens, where it's refracted onto the retina at the back of the eye. And the retina is a multi-layered tissue that covers the posterior surface of the ocular globe. And it acts like the film of a camera in the sense that it detects and reacts to light to produce a chemical signal, which travels through the many layers of specialized cells within the retina that convert it into a neural signal that then travels down the optic nerve to the brain, where it's perceived as an image. And these cells are highly specialized in their formation and function, and they work together very proficiently to produce the detailed vision that we as humans have evolved to possess. And it's important to recognize that these cells don't have any regenerative capacity. So the cells that we're born with serve us for our entire lifetimes from the day we're born into our old age. And so the eye has evolved a number of mechanisms to ensure these cells remain healthy. And the most important of those comes from the choroidal layer of the retina, which is the very bottom layer in this picture. This layer contains many blood vessels which supply nutrients and metabolites to the retinal pigment epithelium layer, or the RPE. And then the RPE exchanges these nutrients to the cells that overlie it. And then once visual transduction processes have taken place, the byproducts of these pathways are then transferred back to the retinal pigment epithelium, which gets rid of this, what is effectively debris, so that it doesn't build up within the retina, and the retina continue to function as normal. So if we were to look at the retina from an on-face view, as in this fundus picture here, the macula is the very central portion of the retina. And this area of the retina is predominantly contains what we call cone photoreceptor cells. So this is a cone photoreceptor cell here. It's a highly specialized cell type that reacts to bright light, and because of the position of the macula, these cells enable us to see centrally, but also in a lot of detail, and they also detect color, so they enable us to have color vision. So there are a number of inherited macular dystrophies that are relatively well characterized. And each of these different conditions are what we call monogenic. So they have one gene which is known to cause the condition. And although they look very different on each of the pictures that I'm presenting here, they do demonstrate what we call heterogeneity, which means that the way that they present within the eye can be very different from person to person. And so just by looking at the retina, it's very difficult to determine exactly which condition a person is affected by. And so it's not unusual for doctors to pursue genetic testing. And due to advances in sequencing chemistries or DNA sequencing chemistries in recent years, we're now able to sequence many genes in parallel. And so this diagnostic process is much more efficient than it used to be. And identifying the exact genetic cause is not just useful for the clinician. This information is useful for the patient because it enables them to understand more about the condition they're affected by, how it might progress to affect their vision in the years to come. But it also enables people involved in their care to understand how the family members may or may not be affected so that they can be monitored more closely. So when we talk about macular disease, most people immediately think about age-related macular degeneration, which is the most common cause of visual loss in the developed world. And AMD is very different in the way that it develops, in the sense that it's a multifactorial condition, which means that it has many different aspects to that lead to its development in an individual. And some of these are what we call modifiable risk factors, so things about our lifestyle that we're able to change, such as our diet, how much we expose our eyes to UV light, and whether we smoke, and to a smaller extent, the amount of alcohol that we drink. These are all factors that affect the health of our retina. And then there are a series of non-modifiable risk factors, so things about ourselves that we're not able to alter. And the most obvious of these is increasing age. This is perhaps the most important risk factor for this condition. But also ethnicity and gender play a part in risk. And what's quite unusual about AMD is that it has a very large genetic component. So there are many changes across, DNA changes across the genome that contribute to an individual's risk of developing this disease. And I'll talk more about that a bit later. So in terms of etiology, AMD develops in the early stages with the presence of accumulations that appear underneath the retinal pigment epithelium and above a layer called Bruch's membrane. And these accumulations, which we call drusen, increase in size and number with time to the point where they begin to affect the function of the retinal pigment epithelium. And as the disease continues, the drusen cause the RPE to die. And in the later stages of disease, there are two different forms. There's dry AMD, which is associated with what we call geographic atrophy, so large areas of cell death in the retina. And then there's also a form called wet AMD, which is associated with a process called choroidal neovascularization, where aberrant blood vessels grow through from the choroidal layer into the retinal space. But these blood vessels are what we call aberrant. They're very abnormal and they're prone to leaking. So patients with this form end up with bleeds within the retina. So in the early stages of AMD research, scientists were very much focused on determining what the contents of these accumulations or drusen within the retina were. in an attempt to try and understand what exactly was going wrong with the biology of the retina. And these studies identified a number of proteins associated with different processes. So proteins associated with inflammation and immune activation, but also lipid accumulation, much like those that we see in heart disease. And perhaps unsurprisingly, given the role of the RP, they also found a lot of components of cellular debris. And so at that time, scientists put forward this hypothesis that an individual's risk of developing AMD was attributed to biological changes that occur with increasing age, environmental exposures that happen during their lifetime. And they also hypothesized that there was a genetic predisposition to the disease, but at that time, due to limitations in DNA sequencing technologies, this portion of the hypothesis was not very well characterized. And that all changed in the early 2000s, when sequencing technologies advanced significantly and allowing us to survey the entire human genome for genetic changes associated with AMD in hundreds to thousands of patients affected by the condition, and by comparing their genetic changes to healthy individuals. And a number of really significant findings were identified through these studies. Perhaps most interestingly, very strong associations were made between genetic variants in genes that are involved in the alternative pathway of the complement immune system. So the complement system is an aspect of our innate immunity that controls the way our body responds to the presence of pathogens. And each of these genes has a role in that pathway, which is really interesting given the finding from the early studies of the contents of drusen. And so despite all of this work and success, it's still not really clear to us how these genetic changes contribute to the development of disease. And so for that reason, genetic information is of very limited clinical use. And currently genetic testing for AMD is not recommended. However, there are some subtypes of AMD that exist called early-onset drusen or early-onset macular degeneration. And these forms of the condition, compared to AMD, which is relatively common in older people, early-onset drusen and early-onset macular degeneration affect far fewer individuals, but they onset at a much younger age. And what's interesting about them is that they seem to have very similar disease pathology to AMD. So this is very unusual. And what is of interest from our perspective as geneticists is that these subtypes of AMD tend to affect more than one individual from the same family, as indicated by this family pedigree here. And so that would imply to us that these conditions have a very strong genetic component, which gives us a rare opportunity to investigate the genetic causes of a condition that could reveal insights into common forms of macular degeneration. And so in the early part of the research I've been doing as part of this program of work, I identified these 6 families. So here on the screen, there are 6 different boxes with different family pedigrees and a corresponding fundus image from an affected individual in each of those families. And each of what we call the proband, so the individual from the family that we begin our investigations on, had gone through the usual genetic testing pathway for ophthalmic or inherited retinal degeneration within the NHS, which now looks at all the genes known to cause retinal dystrophy, but they had not received a diagnosis for their condition from this testing pathway. And so because of their similarities and the fact that there were more than one individual affected in each of these families, we still believe that there was a strong genetic component or cause for the condition in each of these cases. And so we decided to investigate them further. And so I performed an assay called whole exome sequencing, which is a sequencing chemistry that enables us to screen all of the genes within the human genome that are known to make a protein. And the findings were really interesting. So each of these families had a different change in a gene called complement factor H. And this was kind of intriguing to us because CFH, you'll remember from an earlier slide, has a very strong association with increased risk of age-related macular degeneration. But to our knowledge, it wasn't really known to cause what we call monogenic disease, where a patient has a single change in the gene that can be transmitted to future generations and cause them to have disease. So we thought that this was really interesting, and when we investigated it further, we found that CFH in fact makes two different proteins, a long form called factor H and a short form called factor H like 1. And what's interesting about factor H like 1 is that although it's much smaller, it actually retains all of the same functions as the factor H long form of the protein. And we noted at this stage that each of the genetic changes we'd identified in the families we were studying affected both the long form and the short form. But at that time, we didn't really appreciate the significance of this. And then we discussed these findings with another group at the University of Manchester who are interested in the alternative pathway of the complement system. And perhaps conveniently, they'd already investigated the function of this small protein and found that it was expressed almost exclusively within the retina of the eye in humans. And so that enabled us to hypothesize that it was in fact this small protein that was causing the condition we were seeing in the families we had studied. And this information enabled us to design a series of experiments to investigate exactly what these mutations were doing to the protein. And we found from this work that each of the different mutations prevented expression of factor H like 1. And so we were able to hypothesize from that finding that it was loss of factor H like 1 within the retina that was leading to overactivation of the immune system within the eye and causing the development of drusen, likely due to increased inflammation. So because not very much is known about this mechanism of early onset macular degeneration, we've established a pipeline within our laboratory to make disease models of this condition so that we can understand it better. So for this process, we take a blood sample from a patient that we have identified an interesting genetic change in, and we isolate a particular type of blood cell from the sample and grow it in culture within the laboratory. And then we apply a number of reprogramming factors to these cells to convert them into what we call induced pluripotent stem cells. So stem cells are pluripotent because they have the capacity to become any type of cell within the body. And so we're able to exploit this and apply a number of small molecules to direct these stem cells into becoming retinal pigment epithelium. And this is a process that's really important because you'll remember from my first slide that cells within the retina don't have the capacity to regenerate. So it's not possible for us to take a biopsy of an individual's retina to study in the lab to understand the condition in more detail. And so these cells offer an opportunity for us to very accurately replicate the disease mechanisms in the lab so that we can understand what is happening in the disease process. And more recently, we've introduced an aspect of this pipeline where we use genetic engineering in the form of CRISPR-Cas9, which you may have heard about in the media, to circumvent the requirement for sampling patients and growing their cells in culture. We're able to introduce genetic mutations using this genome engineering into healthy stem cells so that we can then turn them into RP and study them in the same way. And this reduces the time that it takes to make RP quite significantly. So once we have RP from these patients, we conduct a series of experiments in the laboratory. So we first conduct a biochemical assay to confirm that the genetic change we've identified does in fact cause a problem in the pathway that it functions in. And then we use a series of proteins to try and rescue this phenotype so that we can doubly confirm that the genetic change we found does in fact cause a problem. And then we're able to study these cells in much more detail using a series of what we call omics technologies to investigate how these changes affect how our RNA is processed to make genes and how this information is then used to make proteins so that we can then understand in much greater detail the biological processes that are disrupted in this condition. So off the back of this early finding, we then recruited a number of other patients with very similar retinal presentations to the families in which we'd identified these CFH changes. And we found that in fact, only around 10% of patients have a damaging change within this gene, which led us to believe that there was other genetic changes that we were missing by just looking at the CFH gene on its own. And we began to wonder what other causes we could be missing. And so we decided to develop a new genetic test to screen for genetic changes in all the genes associated with AMD biology and risk, in order to better understand the genetic landscape of this condition, and in order to improve the care pathways for this group of patients in the future. So, as is often the case when working in research, this has turned out to be slightly more complicated than we first suspected. So we have a lot of experience of understanding the genetics of retinal disease in Manchester. We perform a lot of testing, and for the vast majority of cases, we identify a single genetic change that has a very large impact on the protein that gene makes, and we can say with a relative degree of certainty that it is in fact that change that is causing disease in a patient. And then we're able to follow up that finding with what we call segregation studies. So we're able to track the disease through a family to see if the change follows disease development, or we're able to perform what we call a functional assay, where we investigate the impact of that genetic change on the protein that it makes within the laboratory. And then at the other end of this spectrum is AMD. So our current understanding of AMD is that individuals have a large number of genetic changes with a very small effect. And these genetic changes likely respond or react to environmental factors and lead to the development of disease. But from our work, we suspect that early onset macular degeneration falls somewhere in the middle of this spectrum. So we think that patients who experience this condition have a small number of medium impact changes that may or may not interact with external factors, but significantly predispose them to the development of drusen at a much younger age. And so we hope that by investigating these cases further and understanding which of these changes do have more of an effect on protein function, we'll not only be able to understand early onset macular degeneration better, but it will also inform our understanding of common forms of macular degeneration, such as AMD. So there's a big drive within the National Health Service at the moment to develop personalized genomic medicine approaches to patient care. And what we mean by this term is that we, in the future, will increasingly use a patient's genetic information to inform the type of treatment that they receive. And so this is no less true for AMD. And there are a number of pharmaceutical companies developing gene therapies for AMD at the moment. The two that are most prominent to my mind are a company called Gemini and a company called Gyroscope. And what's interesting about the treatments that they are developing is that they both target the complement pathway or the complement system, which you'll remember from my earlier slides is the pathway that is disrupted in many early onset macular degeneration cases. And so we feel that going forward with increased amounts of genetic testing in this cohort of patients, we'll be able to identify individuals who have a significant genetic predisposition to complement dysregulation, and that this will be valuable information in terms of treatment because it is these patients that will respond most effectively to gene therapy that targets the complement system. And so we hope that through our research, we'll identify further patients that might be eligible for these trials, and that by increasing understanding of the genetic variants that underlie this condition, we'll be able to help these organizations broaden the spectrum of individuals that they're able to offer this treatment to in the future. And that's it. Thank you for listening. I'll be happy to answer any questions if I can.
Brilliant, Rachel. That was very interesting. Loads of new information there. On a number of these talks recently, we've heard a lot about RNA rather than DNA and how that's playing a significant part. So it's really, really interesting. So thanks very much. Jerry, are you about, Jerry?
I am here. Thank you, Colin. Yes. Sorry to be a bit late to the game.
Have we had any questions through?
No, we haven't. But to fill the gap while people are hopefully sort of typing away, I have one from myself for Rachel. What sort of inheritance patterns do you see with early onset macular degeneration? You obviously had several families and then showed the family trees. Is it consistent inheritance patterns or is it vary with mutation?
So the early families that I mentioned, the 6 families that presented at first, they all had dominant disease. So there were individuals affected in each subsequent generation of the family. So that means that they just need one genetic change in the gene. So we have 2 copies of the gene, one that we inherit from our paternal line and one from the maternal line. And so with dominant disease, they would only need to have one gene copy affected to be affected by the condition. That's what we mean by dominant disease. But I think originally in the literature, the first group to describe early onset drusen suspected that it was recessive, meaning that you need 2 mutations in the same gene to have the condition, but this has since been disproven.
Thank you. I'm going to keep going as long as I can because nobody's asking any questions in the chat yet. So how many people do you think have early onset macular degeneration in the UK or in the world? I mean, that was really difficult to guess these things.
It is really difficult. It certainly is quite rare, but what we're seeing is, and one of the problems with identifying this condition is that in the early stages of the disease, it doesn't affect vision. So that in many cases, we don't see these patients presenting to us in clinic until they have much more advanced disease that's beginning to affect their eyesight. But because routine eye testing is becoming much more detailed and thorough, I think we'll start to see increasing numbers of these cases where individuals are found to have drusen at a much younger age. And then I think we'll get a better idea of just how prevalent this condition is.
Thank you. Yeah, okay, a couple of questions from Claire in the chat. Is it possible to have macular degeneration and loss of peripheral vision too?
It is, yeah. So some forms of genetic condition are associated with macular drusen, but patients also get peripheral drusen and what we call retinitis pigmentosa. So that would be then associated with peripheral vision loss also.
Thank you. And a second one from Claire. How would one go about getting involved in clinical trials for early onset macular degeneration? Possibly the ones you mentioned.
Yeah, so the best way to go about that is to undergo genetic testing. So the more people that we can undertake genetic testing on, the more that information can be used to identify people who will be eligible for gene therapy trials in the future.
Thank you. I should mention the gyroscope trials. We're very involved with gyroscope therapeutics and have been helping them to recruit people into their trials for dry AMD at the moment. But we know that they're looking widely, and if they've proven in dry, then yes, as you said, it could help people with early onset macular degeneration as well. So it's very important. And if anybody wants to know more about gyroscope therapeutics, they've got a good website, or they can actually people can sign up to what we call our research participant database as well. There's a form on our website, or we can send out paper forms if you contact us. And it's a patient registry where people can register a few of their details about their eye condition and where they live and so forth. And then when we're asked to help recruit for clinical trials, which is happening all the time, and as I said, we're working closely with Gyroscope, we can identify you and maybe send you information because we think you're eligible and you live close to a hospital that's taking part. So we'd encourage people to look at that. It's on our website in the research section if you want to sign up. Okay, next one from Madeline. If there's a Q&A button, I can't find it. Apologies. I don't think there is a Q&A button.
No, it's fine. No.
Okay, so she says, thank you for explaining gene therapy. I suspect I'm at the end of the spectrum with inherited disease, as my mother also had AMD. My question is about stem cell treatment. I believe so far stem cell treatment can only be used on dry AMD. Is that correct?
That's a very good question. I'm not sure of the answer.
I can answer.
Go on, then, you answer that one.
Well, it's been tried. It's in clinical trials for wet and dry.
Right.
Moorfields Eye Hospital in 2015 treated 2 people who had very severe wet AMD. and we know research is going on and continuing to go on in that field at more fields. But in America, there's a couple of three actually, I think, trials to treat dry AMD. One in particular called OperaGen. They report very regularly on how people within the trials are doing, and they've been showing some really interesting positive results. It's still in early stages, but yeah, I mean, maybe one day this form could treat lots of different types of macular disease. Okay, next one is from Jane. I've had genetic testing, but been told I'm one of the 30% where they have not found the gene. Is there potential that new gene issues will be discovered? And if so, will I need to be genetic tested regularly?
The latter half of the question, to answer that bit first, that will very much depend on the type of genetic testing you've already had. So if you've had a very broad spectrum DNA test, such as an exome or a genome sequencing test, you wouldn't need to be tested regularly, because with your consent, they will have kept that information, and that genetic information will be reviewed as new genes are discovered. And then to answer the first half of the question, yes, a lot of people are very much still investigating inherited retinal dystrophies, and that research predominantly focuses on people who have not received a genetic diagnosis from NHS genetic testing, which is actually very thorough now. So there are still a few patients that don't receive a genetic diagnosis through that testing pipeline, but we are all working very hard to identify new genes all the time.
Thank you. Next one from Edward. Will your study consider patients from other NHS regions?
Yes, I think so. I don't know if it's possible, but if anybody is interested in joining our research study, I'm sure that I'm happy for Colin to share my email address, and I can investigate that further if anyone would like to take part.
Yeah, I can do that. No problem.
I think when we first started talking about your project with us, Rachel, I had a gentleman contact me who had early onset macular degeneration in his family, and I put him in touch with you.
Oh, right, that's great. Well, that's how it should be.
Professor Black was very pleased.
Yeah, that's the whole point of the process, so that's good.
Okay, another one from Nicola. With respect to the rate of deterioration, is there any evidence to show the rate of deterioration increasing as we get older, or is it difficult to predict? In other words, some people remain stable for many years.
Yeah, I think that is true. So I think the rate of deterioration will very much depend on the underlying cause. So we know from the research we've conducted so far that patients who have what we call loss of function genetic changes, which means that the genetic change they have results in the production of a protein that no longer functions at all, they're more likely to have much more severe disease. And with these early onset forms of macular degeneration, just because of the fact that they occur much earlier in life, they do have much more time to progress, and so will eventually impact the visions more severely. It is very difficult at the moment to predict with any certainty how quickly or what the end point of an individual's disease progression will be, because we just simply don't understand enough about many of these conditions at the moment. But hopefully that will change in the future.
Thank you. There's nothing else in the chat function at the moment. People sometimes say to me they feel that their sight is quite stable, but then it takes a dip.
Yeah.
Is that really a sort of sudden progression of the disease, or is it how it's perceived? Because maybe suddenly it's encroaching on a very center of the macula and you're noticing it more.
It certainly could be that. It could also be that because maybe one area of the retina starts to become more affected, it causes the surrounding cells to be affected, and so the area gets larger much quicker. It's kind of like an exponential increase in disease progression. So that might be why they suddenly feel their vision's changed. Yeah, I think that's probably most likely the reason for that.
Okay, thank you. Another question from Edward. Is your study accessing data held on the tested by the 100,000 Genome Project? Are you benefiting?
Yeah. Yes, we are. Yeah. So I am a GSIP member in the 100,000 Genomes Project, and I have been accessing that data to increase the number of people with relevant phenotypes that I'm able to analyze genetic information for, because the more individuals we have information for, the more significant our findings are. So if you are part of that study, I should be able to see your genetic information, yes.
Brilliant, thank you. How are we?
Sorry.
We've finished, come to the end of the questions so far. I don't know how we're doing for time.
We've got a wee while left.
Okay.
Yeah. I do have a question. So one of the questions that we got sent through was about age and clinicians focusing on a specific age for someone to be diagnosed with an early onset rather than age-related macular degeneration. Does age play any significance in this?
Yes, it does, I think. So I think I mentioned earlier, it's very difficult to diagnose this condition when it first onsets, because having very small changes in the retina, such as early drusen, where there are a few drusen scattered around, say, the macula, it may not affect an individual's vision. So determining the precise age of onset is quite difficult in that regard. But if someone presents with drusen in their 30s, that is very unusual. That's much less likely to be the typical age-related macular degeneration. Whereas if you're closer to, say, 50 years of age, I would say that's more borderline. And then anyone over 55, I think, would automatically be categorized by their ophthalmologist as having AMD. I think personally, I feel that if someone has very advanced macular degeneration in their 50s, that would imply to me that their disease onset at a much younger age. It's just that we didn't know about it because it wasn't impacting their vision. So I think it very much depends. And, you know, yeah, it's a difficult thing to define. But yeah.
Maybe some misdiagnosis in some people. I think everybody
possibly. There's a lot of work in the literature about advanced AMD, which, you know, when we look back at that in a few years' time, we'll probably think, well, actually they had early onset macular degeneration, they didn't have advanced AMD. So I think it's just the case that we need to work to understand how these conditions onset in a better way and define it better. And that will just come with more research and finding more people affected in this way at a younger age.
Well, then there's a couple more chat ones I can go to now. Is there evidence that macular vitamin mineral supplements, so the ARID supplements, I imagine, have any benefit for early onset macular degeneration?
I don't think there's been any formal research in that area. I'm not aware of it. But anything like vitamins and minerals and antioxidants even are known to help support the health of the retina. So it would be misleading for me to say that vitamins will mitigate the effects of a genetic condition, but it certainly wouldn't harm in any way. yeah, I think supporting the health of your retina is important no matter what.
Thank you. And another one, can you develop wet and or dry AMD as well as early onset MD? So could you have the age related and the early onset together?
I think if you're affected by a genetic form of early onset macular degeneration, it is more likely to progress to severe disease in the form of geographic atrophy. However, I wouldn't like to say with any certainty that you aren't at risk of developing wet AMD, because I think we just don't know enough about the condition at the moment to understand what genetic changes may or may not lead to wet AMD.
I get the impression there might be quite an overlap between the two, because
yeah,
the
definitely.
wet AMD, which the growth of the blood vessels, happens in lots of different diseases.
Yeah, exactly. And I think the more that we begin to understand about the genetics of these conditions, the more we'll be able to predict those kinds of implications for later stages of the disease.
Thank you. Okay, a few more questions coming through. I was told at 20 that the drusen deposits were like those associated with an 80-year-old, but I didn't get a diagnosis until my 40s. That's from Nicola. So it's not a specific question, but I think I've spoken to people who said, oh, the optician told me I had drusen in my 20s or 30s, but you know, there was nothing that could be done about it. It was just a bit of a curiosity. So I think as maybe opticians are not so aware of it, so they just kind of,
yeah, definitely. I actually had my eyes tested very recently and had fundus imaging done at the eye test, which is now, I think, routine for most people. And I actually discussed with the optician about early onset drusen, and he said to me that he was surprised the number of people that they do detect drusen at a very young age, but they are starting to establish pathways with tertiary referral centres like the Royal Eye Hospital in Manchester and Moorfields in London, so that these people can be monitored more closely. So I think, because until recently we've had difficulties defining these conditions and what they might mean for patients, it's been tricky to know what to do about it, but I think that will change in the near future.
Thank you. And one from Beverly, I was diagnosed with Stargardt's 20 years ago, but have never undertaken a genetic test. Would I or the research of this disease benefit from a test, getting a genetic test?
I think you would certainly benefit from having a genetic test, just so that you can understand your condition better, and that would provide a definitive confirmation that you are in fact affected by Stargardt's disease. I think, as I said in my talk, these conditions are heterogeneous. It's very difficult to sometimes know just from looking at the retina exactly which condition an individual has, and genetic testing provides confirmation of that. And then other people in your family may be interested in knowing the cause of your disease so that they can understand their risk. And yes, absolutely, research always benefits from additional information. So yeah, to answer that part of the question.
And I know of a clinical trial for Stargardt's where you weren't considered for the trial unless you'd had a genetic diagnosis. You couldn't get that genetic diagnosis as part of the trial. So if you're interested in potentially taking part in clinical trials, getting that genetic test would help along.
Yeah, exactly. All the information that we gain from genetic testing, we always keep a record of it should we be contacted in the future about the opportunity for patients to participate in clinical trials, just like you mentioned with your database, Geraldine. We can do that as well.
Okay, a good one here. Should we give up booze? Hopefully not. Everything in moderation.
Yeah, exactly. Everything in moderation. The link between alcohol and retinal degeneration is not as significant as getting old, so I'll leave it at that.
Okay, a couple couple more if we've got time.
We've got, yeah, time for 2 more, I think.
2 more. Okay, I might have to be selective then. I was diagnosed in my early 40s only when I had a change in part of my vision. I had suffered from extreme light sensitivity since my late teens. Is it likely that if I had had fundus imaging prior to my 40s, that I would have received an earlier diagnosis? This imaging was not offered until I had an issue.
That's quite difficult to answer, really, because sometimes there can be changes occurring in the retina that we don't necessarily see on imaging. But certainly symptoms of light sensitivity is something that should be brought to the attention of an ophthalmologist, especially at such a young age. Yeah. There is, I mean, it's difficult to answer that question with any certainty. But yes, there is a possibility that that lady would have received a diagnosis earlier if genetic testing, for example, had been pursued when she was experiencing light sensitivity, rather than just following a visual change in her retina.
The imaging that we have available to us now is so much better than it was 10, 20 years ago. Yeah, it's difficult to say. Okay, last one. Do we have an indication whether the genetic factors for AMD are more prevalent and passed on from mother to daughter, or mother to son, or father to daughter to son? I think it comes back to that inheritance pattern, doesn't it?
Yeah, so there are some genetic changes associated with quite a large increased risk of AMD. And we know that they are passed from generation to generation, irrespective of gender. And what we are starting to see now is that there is an even bigger risk to that individual if they inherit 2 abnormal copies of that genetic change. So say one from their mum and one from their dad, compared to someone who just has 1 copy of the genetic change. So hopefully that answers that question. I think, you know, there is a lot that we don't understand about the genetics of AMD at the moment, and that's why genetic testing isn't recommended, but we're, through our research, trying to change that so that we can understand the heritability of the condition.
Thank you. Have we got time for one more, Colin?
Yes, please. Yeah, I think another
Thank
one.
you. Somebody said they really want the answer to this. Does early onset macular degeneration present alongside myopic macular degeneration?
Myopic macular degeneration is something that's slightly different from my understanding. But that's not to say that you know, myopia is quite common. So you could potentially be misdiagnosed with myopic macular degeneration. I'm probably not the right person to answer that.
Don't worry.
Don't worry.
Yes. So yeah, from what my understanding is, myopic macular degeneration, because of the eyeball is the wrong shape, it puts a lot of pressure on the back of the eyeball, and it just, by the time you reach middle age, the pressure and the stress on the eyeball can lead to all sorts of things going wrong with the macula, with the retina. It's not, I mean, there's a genetic component, I think, to getting severe myopia, but it would probably be different to what would the impact of early onset.
Yeah, exactly.
Not sure.
Excellent. Well, there we go. I think that's drawn us nicely to a close. Rachel, thanks very much. There's been people are saying some very nice things about your answers and how clear they were. So thanks very much.
Thank you. I'm really happy about it, so thank you.
So my iPad is now waffling onto itself. I can hear the computer voice and trying to talk over it. It gets all a bit bizarre. Anyway, well, thank you ever so much for joining us. It's been a really good session. Certainly learned a lot. The video of this will be available on YouTube in the next week or so. for if you just want to have a recap and have a watch. Rachel, I'll send the link to you so you got it yourself. Jerry, you mentioned the database for our database of research cohorts, so that's really cool. So all it leaves for me to do is just say thank you all for coming and taking part. There's another session at the last Thursday in August, which is on cone dystrophy with Professor Andrew Webster from Moorfield. So that should be another good session. So other than that, have a really nice evening. I suggest having a glass of wine, but as Rachel says, in moderation. And we'll see you on the next session. Thanks very much. Bye.
Thank you. Bye.
Thank you.